Hi everyone, Dr. Hinkey here with our video lecture on Chapter 4, Cell Structure and Function. Now you've completed this lab and so this should be pretty familiar, but we'll go over it one more time. There are some parts that always need some clarification, especially differentiating between our prokaryotes and eukaryotes. So this is the chapter outline. We'll go over the cellular level of organization, exactly what that means. We'll look at prokaryotic cells, compare those to eukaryotic cells. Then we'll go through some specific parts or compartments that are found in eukaryotic cells, some structures that help our cells out, that are parts of our cell and what they do. And we'll talk a little bit about how these work together. So cellular level of organization, remember we started out with atoms, we brought those together to get molecules, we bring those together to get our biological macromolecules that come together to make up the organelles or the little parts of a cell. But until they all come together in a specific manner, the cellular level of organization to form a cell, they are not living. They don't have those 6 properties that we defined as being what has to exist for something to be considered living. So one of the unifying concepts in biology is the cell theory. And I really like, this is a great example. We've talked about the scientific method and we talk about the difference between a hypothesis and a theory. And if you watched the video where I use a puzzle as an analogy to the scientific method, remember the whole puzzle is the theory. The hypothesis would be any one part of that puzzle. It's one piece of the whole big picture that we're trying to build to understand something in the world. So the cell theory, we have 3 basic statements, but if we really look at the complete and comprehensive cell theory, there are about 72 statements. But these 3 really give us our foundation that we've built on to have our puzzle. So in the cell theory, remember a theory is a predictive model that we've derived from lots and lots of different hypotheses on a related topic. So our related topic here is the cell, the basic unit of living things. And people have proposed and tested lots and lots of hypotheses and experimented and gathered more and more data and information and tried to disprove things. And the more they can't, the more evidence we have that it is correct. But we're not going to say we proved it because we can always modify and revise it. But once we get to the level of a theory, what we have is enough information, enough pieces of the puzzle in place that I can accurately predict what's going to happen by looking at this puzzle. So even though pieces are missing, I am confident that what I do have is correct, and I can make some predictions about what's in those places where I don't have the pieces in place yet. So a theory doesn't have to be complete. We will never completely and fully understand the world around us or run out of questions to ask. So we're continuously building. That puzzle is constantly expanding as we come up with new hypotheses to test. But our foundation, our predictive value, the model that we have tells us that all living organisms are composed of cells. No exceptions have been found. This is one of the reasons that viruses are not defined as living, is because they can't carry out those 6 different properties or characteristics of life on their own without being in a host. They don't have the cellular level of organization. They have bits and pieces of a cell. They have a protein coat. They have a nucleic acid, but the rest of the parts and pieces are missing, and so they can't metabolize on their own. They can't reproduce on their own. They can't do all of those other, they don't maintain homeostasis on their own. They need a host cell to live in and provide their energy, their resources to build new ones, to replicate everything else. So nothing is considered living until we get to the cell level of organization. The second statement in the cell theory is cells come only from pre-existing cells because cells are self-reproducing. So all cells come from pre-existing cells. This idea of biogenesis. So genesis is the creation of life only from other life. Louis Pasteur disproved the hypothesis of abiogenesis, that life randomly or spontaneously arose just from the matter around us. So we know that cells come only from pre-existing cells through cellular reproduction. Cells are the basic units of structure and function in organisms. So our body, composed of 35 trillion cells, more or less, depending on how big you are, but somewhere in that range is average for an adult human. All 35 trillion cells work together to form those higher levels of organization, our tissues, which then form organs, which work together to form organ systems that make individuals. But every one of those previous or subsequent levels is built on our previous level, this level, our cells. And some living organisms are a single cell, and others, like us, are multicellular, all of those parts working together. So we'll look at our simplest cells first, prokaryotes. And I'll tear apart these words a little bit because prokaryotes, eukaryotes. We get some confusion. We had some difficulties in the top hat on the labs differentiating these two. If we look at these words, this prefix pro means early or before. The prologue of a book is presented first before the story starts. So before. And EU, this prefix is Greek, means true. Karyote, this is Greek for nut or seed. And when early scientists with their primitive microscopes looked at these cells, they could see some that, hey, it looked kind of like if I cut a peach in half, there is a nut in the middle, the seed in the middle. And others don't have that. And so they divided cells into eukaryotes, true seeds, and prokaryotes, before there was a seed. And now we know that karyote, that seed, is the nucleus, which we can see. So our prokaryotes are before that evolved. They came before that. We have no nucleus. So our prokaryotic cells do not have a nucleus. They are much smaller and simpler than eukaryotes. I like to compare these. So when we say they're structurally simpler, our prokaryotes have one big room where everything happens. Our eukaryotes have these membrane-bound organelles inside. They have compartmentalization. So a prokaryotic cell is like a one-room schoolhouse. A eukaryotic cell is like a big school with lots of different classrooms, a separate place for administrative functions, separate places to eat. So everything, all the activities of the day are divided into different places versus prokaryotes, these simpler cells, one-room schoolhouse, everything goes on in the same place. Right? So the teacher grades the papers, the administrative duties are carried out, teaches the class, and then the students eat all in the same place. My prokaryote is the simpler, smaller one. My prokaryotes are divided into two domains, the bacteria and the archaea. Until fairly recently, these were in the same domain. They were all together as just prokaryotes. But as our technology increased and enabled us to look down at the molecular level, what they're made up of, we found there are significant differences between the bacteria and the archaea that led to their being divided into two different domains, and our third domain is eukarya. Some of the differences are because archaea have ribosomes that are different from bacteria. They're not eukaryotic, but they're a little bit different from our bacteria, enough so that they are maybe the intermediate between these two groups. They have some different cell wall from bacteria. So they have pseudopeptidoglycan instead of peptidoglycan. But molecularly, they are distinct enough that they have been divided into their own group. In microbiology, for an environmental microbiology, the archaea are a really, really important group. We call them extremophiles because we tend to find them in extreme habitats, the very, very bottom of the ocean where the pressure is extreme, in the deep sea hydrothermal vents where temperatures are 300 degrees Celsius, in just really, really crazy in places with no oxygen, in places with high methane, and places where we would propose in the Arctic, in Antarctica, propose, nothing could possibly live there. And then we find these guys. To date, none of the archaea have been found to cause disease. But again, as our tools and our abilities to identify these become better and better, and our knowledge advances, we may find they do. So especially now, these were typically not associated with humans, but we might have some of these in our gut biota, so living symbiotically with us. We have lots of bacteria living symbiotically with us. And here we say they're smaller than eukaryotic cells. So I mentioned our bodies are made out of about 35 trillion eukaryotic cells living on us or in us symbiotically, mutualistically, benefiting us as we benefit them. We have 10 times as many bacterial cells. So bacteria, good. We would be very unhealthy if we didn't have our symbiotic bacteria living on us or in us. But that just gives you some perspective of the size, that we can have 10 times as many of them living on us and in us, and can't see them, don't know they're there. They're that much smaller than our cells. So these two are structurally similar, but they are separate domains. We're going to really focus on the bacteria. So look at the structure of prokaryotes, of our bacteria. Under the microscope, they're very, very small. This would be tremendously magnified. You would see little tiny dashes under the microscope at the highest magnification, oil immersion. Little tiny circles. Some can be fairly big, but when you look under the microscope, you're not going to see parts. You're not going to see a nucleus. You're not going to be able to identify specific parts as you can with many eukaryotes. But because they have a cell wall that gives them structural support, it's this rigid cell wall, they maintain shapes. And so the shapes they come in are coccus, or cocci in the plural. So you've probably heard of staphylococcus or streptococcus. Coccus means that the cells are spherical. And then staphylo means they're in clumps or clusters, and strepto means they're in a chain. So this is some streptococcus. Bacillus means rod shape. So bacillus, or bacilli, plural, are rod-shaped bacteria. E. coli is an example of a rod-shaped bacteria. And then we have the vibrios, or the curved bacteria. We have some that are just a simple comma. We don't have a vibrio shown here. But the vibrios include things like the causative agent of cholera, vibrio cholera, which is just a comma. Or we can have spirochetes, which are corkscrew shaped, and spirillum, which again, a little helical, but flexible versus rigid. The cell envelope, or the thing that separates the inside of any cell from the outside, called the cell envelope, the plasma membrane, is the primary one. This is all cells have a plasma membrane. This is a phospholipid bilayer. Remember our phospholipids. that phospholipid bilayer with embedded proteins. All cells have that plasma membrane, and it is the definitive structure that determines here is inside the cell, here's outside. The plasma membrane, because they have the consistency, they're phospholipids, they have the consistency of olive oil, they can infold, can increase surface area. This is important in all cells, but in bacteria, it's really important because those proteins that are embedded in the membrane carry out all the metabolic functions, or most of the metabolic functions in bacteria. Those internal pouches can be called mesosomes. Meso is middle, bodies. And we're going to look at why those mesosomes are important to eukaryotes, because that's probably where our internal structures came from. The cell wall maintains the shape of the cell, as I mentioned. It's peptidoglycan in bacteria. Glycan means sugars, peptido, amino acids in chains. So it's made of alternating layers of sugars connected with amino acids. That's in bacteria, my prokaryotes in my bacteria. Eukaryotes have cell walls that are made of cellulose if you're a plant, or chitin if you're a fungus. Both of those are complex carbohydrates, are polysaccharides. So the bacterial cell wall is made of a different substance, and it's what gives shape, just like our plant cells and fungal cells can have a specific shape because they have a wall. Animal cells just have that phospholipid bilayer, so their shape's a little more fluid. And then a glycocalyx. Not all bacteria can produce a glycocalyx, but some do. And what that is this external layer of polysaccharides. They basically put this jelly-like coating around the outside for protection. It hides them. If my immune system cells are looking for a bacterial infection and they come over and say, hey, what are you doing here? And they reach out and touch this invading cell, they'll say, oh, you're a polysaccharide, you're food, I don't care about you, and they'll leave it. So it's a way for bacteria to hide, to get into our bodies and hide from our immune systems to cause infection. And it's sticky, so it also lets bacteria stick together. And this is the basis for biofilms. You're probably familiar with biofilms, some of the places that they form. If you have a dog and you don't wash that water dish out all the time, it gets nice and slippery on the inside. That's the formation of a biofilm, of bacteria sticking together with their glycocalyx, forming this nice film on the inside of the bowl. That's also the beginning of, if you have a boat and the bottom starts to get slimy and lots of algae and things stuck on it. All the algae, all those things can't grow until I have a biofilm on the outside. So that starts that fouling, that biofouling of vessels starts with a biofilm made of bacteria. And the one everybody's probably most familiar with is bacteria in our mouth will form biofilms on our teeth. So if we don't floss, we end up with plaque, which is a big biofilm. So we'll look at prokaryotes, at the structures that we have in them. We'll just go through kind of quickly. So all cells have cytoplasm. That's the aqueous solution. It's a liquid inside the cell. It's filled with all sorts of dissolved substances. This is where all of the resources that we need to function, to build new matter. So when we're going to divide into two cells, we need more materials. So we store our materials in here, our nutrients, all dissolved in this aqueous solution. I have my plasma membrane because all cells have a plasma membrane. And you'll see this is inside, it's the inside layer, inside of my cell wall. So my cell wall is the covering, the rigid structure outside of the plasma membrane. And then if a bacteria is able to form a capsule, that'll be on the very outside. Not all bacteria can form a capsule. Many can, but not all. They all have a cell wall. All cells, prokaryotes and eukaryotes, have a plasma membrane and cytoplasm. All cells, prokaryote and eukaryote, have DNA. This is the instructions for how to live, for what they do, to perform their functions, for metabolism. All the instructions are in that DNA. In bacteria, that DNA is one single ring of DNA, but it's just floating around in there, so it gets all tangled up into this mass. And where the mass is located is called the nucleoid region. This is an aqueous solution. That nucleoid region can move. It is not bound by anything. It's just floating in there. So it's not a set region, but if I have the ability to look closely enough, if I have the right kind of microscope, and I see the darker, the denser region inside the cell, I know, that's where all of the DNA is, where that single chromosome is. So that's my nucleoid region. And ribosomes. So all cells, eukaryotes and prokaryotes, have ribosomes. This is a site of protein synthesis. This is pretty critical because metabolism, the sum total of all the chemical reactions in a cell, every chemical reaction is sped up using enzymes. Where enzymes are my class of proteins that are used in metabolism to make sure that chemical reactions happen at the rate that I need them to sustain life. So I need to be constantly building those proteins, those enzymes. So these are really important. We find them in all cells. So all cells, cytoplasm, ribosomes, DNA, and a plasma membrane. Prokaryotes specifically have a cell wall, which we have some eukaryotes with cell walls, but not all. We don't have a nucleus. We don't have a house for our DNA in a bacteria, so just that nucleoid region. Inside, we don't really have a whole lot of other things. If you see here, we can see, follow that color, this infolding of the membrane. This is a way, that infolding, these mesosomes, this infolding is a way for bacteria to bring foods in. So things have to get through this outer capsule and the cell wall, and then they're brought into the cell through the membrane. We'll talk about membrane transport coming up shortly. So our outer coats are permeable. They are some things can get through them, some can't, but some things that the cell needs are able to get through. It is not fussy about what can get through. That's pretty much a function of size. Is it small enough to fit between the molecules? Sure, then it can come in. All right, and the capsule we talked about, some have a capsule, some don't. So that's the outside. Or the inside, we've got, as we move outside, we see all these little threads. Not all bacteria have these. These are called fimbriae. And the fimbriae are these hair-like bristles. They look like cilia, but they're not. These aren't used for motility. They're not used to move. They don't sweep like oars like the cilia do. These are just hair-like bristles. They make our bacteria fuzzy. This also allows for adhesion. It lets our bacteria catch on to things and stick. So if two bacteria are floating past each other, their fimbriae can get tangled up and they'll stick. So we can also form a biofilm with these. These can also, a lot of bacteria that infect our upper respiratory tract have these, because our upper respiratory tract cells have cilia, little beating bristles that beat. They're usually used for motility. Our respiratory cells aren't moving, they're stationary, but they're constantly sweeping mucus up. So when we get sick, we'll produce mucus and sweep those cilia upward to remove bacteria. We'll produce that mucus to capture bacteria, and those cilia will sweep upward. Well, those cilia look like this. If a cell with fimbriae gets into your respiratory tract, these get all tangled up in the cilia, and it makes it harder for us to remove this bacteria. Some bacteria have flagella, not all. So some have this long whip-like tail. Bacterial cells can have all sorts of flagellar formations. They might just have a single one here. They might have one here and one at this end. They might have 3 or 4 or 5 of them at one end. They might have flagella all the way around. So they have lots of different flagellar configurations, whereas most eukaryotes either have one or one at either end. The other structures that some bacteria have, they may have a pili. And we can sometimes see that the pili is referred to as the conjugation pili. And what the pili are used for is for conjugation, not sexual reproduction. Bacteria have this ability to share bits of DNA. So two neighboring bacteria, not share as in I'm going to reproduce and replicate and go from a parent cell to a daughter cell, not passing it on that way, but hey, neighbor, look, I have this little gene here that makes me resistant to an antibiotic. Let me make a copy for you and pass it over to you. And what the pili does, it's very similar to a fimbriae, except it's a little bit longer. Kind of acts like a hook, grabs onto a neighbor, reels it in till they're touching, and then passes that bit of DNA to the neighbor. Those bits of DNA that they pass are typically extra chromosomal pieces, but they can pass pieces of their own DNA, their chromosome. This is what they need to function. Plasmids are these little accessory or extra chromosomal rings of DNA. It's like bonus DNA. They don't need it to survive. It's not instructions that are essential for the organism to live, but it usually confers some sort of benefit, so they keep it around. And then they might make copies to share with other bacteria. So those are our prokaryote structures. That's pretty much all we need on our prokaryotes. So we'll move on to eukaryotes. So our eukaryotes, just like our prokaryotes, have DNA because all cells have DNA. They have ribosomes, cytoplasm, and a plasma membrane. So all cells have those. What extra stuff do eukaryotes have? Well, they have a membrane-bound nucleus. So we have a plasma membrane, it's called the nuclear envelope, that separates out our DNA from the rest of the cell. And that's going to protect our DNA. That's going to make sure it stays intact, that it doesn't get interfered with other chemical reactions, with things that are going on outside of the nucleus. So we give it its own place to reside. And then we also have all these other membrane-bound organelles within our eukaryote. This is what separates or compartmentalizes different functions of the cell in different places. And this is what lets us go from being a single schoolroom house to, say, any of the buildings on campus at Trident Tech. Ribosomes are organelles, so we can't say that bacteria don't have any organelles, but they don't have membrane-bound organelles because our ribosomes don't have a membrane around them. They're just made of protein and RNA. So how did we go from prokaryotes to eukaryotes? How did we get those organelles that let us have a bigger size? The chapter talks a little bit about limits to the size of a cell and its surface area. And so having all those membrane-bound organelles in our eukaryotic cells gives us more surface area, more place for chemical reactions to take place. So we can get more efficient, and we can also get larger because we have more surface area. So the Endosymbiont Theory describes how this came to be. How did we go from these simple single cell, no organelle prokaryotes? And the Endosymbiont Theory looked at those mesosomes. So researchers said, when these fold in, sometimes they fold in these real convoluted ways and then pinch off and leave behind membrane-bound sacs or vesicles. And sometimes when they pinch in, they grab onto other things. while they're pinching in and folding up and grabbing on. And then they house separate things. It gives me all this surface area where more chemical reactions can take place. Remember, that membrane has proteins that are enzymes, so chemical reactions take place there. So that allowed this cell to get bigger and bigger. And because sometimes when this is called invagination, when it folds in, sometimes when that happens, it grabs onto something. Well, it could potentially grab on to the DNA or engulf all the DNA. That would be a big benefit to a cell because that DNA would not be interfered with in these other reactions. So as we got more and more interior space, more and more of this protection of this separation of functions, those cells would have had a benefit. So because they could get bigger, because they could do more things, because their chemical reactions wouldn't have to be separated out by time if they were separated by place, because we don't want our reactions to interfere with each other. So that would let us get bigger and bigger. Then in the 60s and 70s, a researcher named Lynn Margulies proposed the Endosymbiont Theory. So that gave us our membrane-bound organelles inside. But the Endosymbiont Theory took it a step further and said, you know, the chloroplasts in plants and the mitochondria in animals and plants look like bacteria. They're the same size as prokaryotes. And bigger prokaryotes would eat smaller prokaryotes. We know that. That happens all the time. We can engulf them and eat them. So what if a big prokaryote, a big single cell, engulfed a smaller one, but then something failed during digestion? And all it did was engulf it and hold it inside. Well, this bacteria is now safe from predation, and it's just going to hang out and do what it does. It's going to eat. So whatever this bigger one eats, it's going to get some of those nutrients to keep eating. Because it's wrapped up in the membrane that engulfed it, so we can see here it being engulfed, this bacteria would now have two membranes. And maybe it just stayed inside there and did what organisms do when they eat. It produced energy. It released ATP. It released energy that would give this bacteria here a big advantage to let it grow more and do more things. And similarly with the chloroplast, if a bacteria engulfed a chloroplast that was able to, or a bacteria that was photosynthetic, and that bacteria would now have two membranes, it would be able to rely on the photosynthesis from this bacteria to get glucose. Wouldn't have to eat anymore. It could just use this guy to get all its food. And of course, this was the 50s, 60s, early 70s, and Dr. Margulies, female, and got a nice condescending pat on the head at the time, until we got the molecular techniques in the 80s that said, oh, hey, guess what? Mitochondria have the same ribosomes that bacteria have, which are different from the ribosomes that eukaryotes have. Chloroplasts have prokaryotic ribosomes, not eukaryotic ribosomes. So our ribosomes are different, and the ribosomes in our mitochondria and our chloroplasts are different. Then we got better and better techniques, and the proteins on the membrane surface are similar to bacteria, not to eukaryotic proteins. And then we got even better technology, and it turns out mitochondria have their own chromosome. They have their own DNA, and it's different from the DNA in the nucleus of the cell. It's a single ring, just like in prokaryotes. Same thing in chloroplasts. So all that evidence together supported Lynn's hypotheses that our mitochondria and our chloroplasts started as a symbiotic relationship that benefited both of these organisms. And eventually, over evolutionary history, over millions of years, that partnership became so firmly established that the mitochondria are no longer a free-living organism. They are now part of the eukaryotic cell. Same thing with chloroplasts. So all of that support, continuing to test those hypotheses, led to so much evidence that now the endosymbiont theory is completely accepted in the field of biology. The evidence, as people look more and more closely at mitochondria, at chloroplasts, we see, yep, that's pretty much where they came from. We'll talk about mitochondria some more coming up, and chloroplasts when we get to photosynthesis and cellular respiration. So let's dive into our animal cell and our plant cell anatomy. And I'm just going to go through fairly quickly what each of the organelles does, because I'm going to put it all together in a story. So first here we have our nucleus, which is a phospholipid bilayer that's all membrane, housing our DNA, which we store in the nucleus as chromosomes, which are long thread-like structures made of chromatin, DNA and protein, as opposed to the single circular chromosome of bacteria. Also inside the nucleus, we have a region that's even darker and under the microscope, more densely packed with molecules. This is called the nucleolus. This is the region in the nucleus where we're making the subunits of the ribosomes. So I said ribosomes are made of RNA and proteins. So right here, we're making those RNAs, the ribosomal RNA, that can go out here and wrap around some proteins to become ribosomes. And my DNA is in here. My envelope, my nuclear envelope, we call it the nuclear envelope, same phospholipid bilayer as all of my membranes, but it's a little bit different in that it has these pores, it has these fairly large openings so that RNA can get out. If we see this membrane system connects to this next structure right outside of the nucleus and connected to it, so this envelope continues into these flattened vesicles, this whole series of flattened vesicles extending out from the nucleus. This is called the endoplasmic reticulum. And the endoplasmic reticulum, you can see, is dotted with these little yellow dots. Those are ribosomes. And ribosomes are the site of protein synthesis. So my endoplasmic reticulum, this is known as the site of protein and lipid metabolism, or protein and lipid synthesis. This is where I build proteins and lipids. The way I do that, well, ribosomes are the site of protein synthesis. The other thing the endoplasmic reticulum does is it packages and transports these. It moves proteins and lipids that are being built around the cell. So the protein portion, when I build proteins, what I do is I take my DNA and I make a copy called RNA. I don't copy all of the DNA. If we think of the DNA as a recipe book, as a cookbook, if I'm going to cook something, I only need one recipe, not the entire book. So out of all the recipes in my DNA book, I am just going to write out the one I need on a note card, on an index card. That's my RNA. It's the single recipe. That comes out here to the kitchen, which is the ribosome, where I make my proteins. So I take that recipe and I make it. And what I have a recipe for is proteins. And then I store those proteins. So here we see this blue at the tip. These are proteins that I've made. I make them as that RNA passes through the ribosome. That protein moves into my rough ER for storage. Some of those proteins are just proteins that I need. Some are going to be enzymes that I need elsewhere. Any protein that I need, I'll make in here. Then if I'm going to make lipids, what I'm going to do is I'm going to make proteins that are the enzymes that carry out the chemical reactions to build lipids from parts, from the phospholipids, from the fatty acid tail, from the phosphate group, from glycerols. So I would make those enzymes. I would take them into my rough ER, and then I would transport them. I could break off. We see this end is sort of pinching in here. Remember, it's the consistency of olive oil, so I can break off little bubbles. So I can break off a little bubble. Here's one that's broken off here, and I can move that to where it's needed. Might be needed in my smoothie art so that I can build the lipids, and I'm going to make my lipids here. And then I'm going to package those in my smoothie ER. So rough ER, I'm going to synthesize and package and transport proteins. Smoothie ER, I'm going to synthesize, package, and transport lipids. Where I'm going to transport them to is this series of flattened sacs called the Golgi apparatus. So the Golgi apparatus, it's named after the guy who defined what it does, Dr. Golgi. These little transport vesicles that have broken off from my endoplasmic reticulum can come and merge with my Golgi apparatus. Remember, phospholipid bilayer has a consistency of olive oil. I could put two bubbles of olive oil together and they'll merge. So I merge and I come in here, and what the Golgi apparatus does is it processes and package and secretes modified proteins. So whatever molecule I need to move out of the cell, or that I might need to replenish my plasma membrane, right? So my plasma membrane is proteins and lipids. So I'd process them here, get them to their final shape, and then again, break off a little transport vesicle bubble and send that outside. And here I merged with my membrane and I'm releasing this molecule. So that's the endoplasmic reticulum and the Golgi apparatus. Let's see. Some other things that I can make and break off from my endoplasmic reticulum are little garbage vesicles. So some of these, a transport vesicle is just either of these membrane-bound organelles breaking off a little bubble that's carrying something. So that's a vesicle used to transport things. Lysosomes are specific vesicles that the protein they carry is an enzyme that breaks down cellular debris. So sometimes in this process we make mistakes, or we get toxins, or we get something that doesn't belong or doesn't work right. We need to break it down and get rid of it, our cellular garbage. And so these lysosomes will digest and get rid of the garbage. So lysosomes are the garbage cans. Peroxisomes are a similar type of vesicle. They have a similar function. They're involved in getting rid of toxic wastes from aerobic respiration. Polyribosomes, we'll talk about this when we get to protein synthesis. My centrosome. So I have centrosomes and centrioles here. If you look closely at this picture, these look like two barrels that are perpendicular to each other. When I have those two barrels perpendicular to each other, they are called the centrosome. Each barrel is called a centriole. These are cylinders of microtubules, and together they organize and arrange all of the different microtubules that make up the cytoskeleton. And the cytoskeleton, these are a series of different sized proteins that maintain the cell shape, and they help things move around. So cell doesn't really have a permanent hard skeleton like we do. I can keep making and extending these little proteins to move things around. Think of them sort of as a railroad track. And it's made up 3 different types of filaments. So those are my parts of the cell, of a eukaryotic cell. And we'll spend some more time on many of these, but I'll give you a good idea. My plant cell, some of the main differences. Plants have all of these organelles. So I have my nucleus, my nucleolus inside there, my rough endoplasmic reticulum, my smooth endoplasmic reticulum, my Golgi apparatus, my mitochondria. I mentioned mitochondria here. Animals and plants both have mitochondria because mitochondria are the site of cell respiration. This is where I get the ATP, the energy my cell needs to function. All cells need energy to function, and they need energy in the form of ATP. It's the only energy form they can use. We'll talk more about that coming up. Plants also have chloroplasts. They have that cellulose cell wall we talked about. The most obvious feature is this nice big central vacuole. So while I can have transport vesicles and vacuoles that hold liquid or hold substances in my animal cell, I don't have this one that dominates the whole cell. The central vacuole in plants is used to regulate water when water comes in and goes out. I have this pretty rigid, solid cell wall, so it makes it kind of hard to get things in and out of the cell, maintaining water pressure. This gives me a little water storage capacity, and I can get rid of some of my toxic wastes by putting them in there. Get them out of the cell. Well, it's hard to get through that cellulose cell wall, so I'll just store them in there until I can break them down. So those are the main differences in plant and animal cells. Oh, and animal cells do not have those centrioles. They have a different method for organizing their cytoskeleton. They don't need quite as much of the structure of the cytoskeleton as animal cells, because remember, without a cell wall, this is the consistency of olive oil. So this cell would be very loosey-goosey, mushy. So we have cytoskeleton filaments just underneath this membrane that help to give it some shape. They can also be arranged as railroad tracks to help organelles move. And I think I'm going to get to that in a minute. So my endomembrane system, talk about the endomembrane. That's those organelles that all have the membrane around them that are all sort of connected together and work together. The nuclear envelope, the endoplasmic reticulum, the golgi apparatus, and those vesicles that can break off from that. So here we have my endoplasmic reticulum, and typically this is how we find it, is adjacent to the nuclear envelope, an extension of it, with the rough ER closer to the nucleus and the smooth ER further out from there. And then even further out from that, closer to the outer membrane, the plasma membrane, we have the Golgi apparatus. So, endomembrane system story, just we usually learn all the parts of the cells, memorize how they work, don't really have any clue how they work together. So give you an example so you can remember how they work together, of a cell making something. So in the endomembrane system is used to make things that are either going to be used by the plasma membrane, so those phospholipids and the different proteins in the membrane, or they're going to make particles that are going to be released out of the cell, so cellular secretions. I have ribosomes scattered throughout the cytoplasm as well. So if I have to make proteins for chemical reactions that are going to take place in the cell, I'll use those ribosomes. For the endomembrane system, I'm going to use the rough ER and the ribosomes attached to it. So my example that I'm going to give you is about milk. In mammals, the mammary glands make milk. Well, it doesn't appear by magic. Cell doesn't go to the grocery store and pull it out of the cooler. We have to make the milk. So in my nucleus, I have my DNA with my recipes for everything my cell could possibly need. I only need the recipe for milk. So I'm going to transcribe that DNA. I'm going to write it on a little note card called RNA, and I'm going to send that out here to a ribosome. What's milk made out of? So think about the parts of milk. What's a protein? So it has protein, casein, is milk protein. It has sugar, lactose, and it has lipids, because milk is fatty. So I need all those three parts if I'm going to make a milk molecule. So my recipe card has the recipe for the proteins, casein, that are in milk, and I'm going to come here and I'm going to make that. And I'm going to make the enzymes that are going to let me build the appropriate lipids. And then those enzymes will come to my smooth ER where I will build the lipids. The proteins that I made here are going to be transported to the Golgi apparatus. The lipids will be transported to the Golgi apparatus. I have enzymes that are going to help with lactose. If I don't have the sugar lactose, I can use carbs, carbons, hydrogens, and oxygens. I can use other types of sugar and modify those with the right enzymes into the sugar that I want. Here, my Golgi apparatus. So I've moved my parts to the Golgi apparatus using transport vesicles. Golgi apparatus is going to package and process those molecules. It's going to take my milk fat, my milk proteins, my lactose, and it's going to put them all together into a milk molecule. And then the Golgi apparatus will transport that with a transport vesicle out to the plasma membrane. It'll merge with that plasma membrane and expel the milk particles into the mammary gland, and baby gets fed. So all of those components work together. If there's a mistake and I make a bad milk molecule, one of those lysosomes that I made over here by putting some digestive enzyme into a vesicle and released it out here is going to find that bad particle and merge with it and digest it and break it down into its building block schnapps and discharge those out of the cell to be removed with our circulation. So that's the endomembrane system and how those components work together. Vacuoles, these are sacs, membranous sacs that are larger than vesicles. They store excess material. The central vacuole in plants is the big one, and we distinguish that from contractile vacuoles in protists or from other vacuoles in eukaryotes, because it's the big giant one that's used specifically for water control in plants. We always distinguish that by calling it the central vacuole. So there's our central vacuole. We have our energy producing organelles. We're going to spend a lot of time on these when we get to photosynthesis. So I'm not going to talk about those a lot here. We'll get a close-up view of these and talk about what goes on in each part here. When we get to photosynthesis, this is where photosynthesis, where plants take carbon dioxide and water, and through photosynthesis make their glucose. So plants are autotrophs, self-feeders. They make their glucose, which will then go to the mitochondria to be converted, broken down to release ATP, the energy for the cell. And then the mitochondria, same thing when we talk about cell respiration. We will look at mitochondria. Both plants and animals have mitochondria because for cells to do work, they need energy in the form of ATP. So here is where we make our ATP. If we're an animal, we eat our food, and then we move the glucose here to be processed through cellular respiration to give us ATP. If we're a plant, we got our glucose through photosynthesis, and we're going to move it here so that we can break down that glucose to get ATP. The cytoskeleton, we've got 3 types of filaments that are involved in moving things around in cells. Actin, this is one of the proteins that we actually find in muscle fibers. It's also part of our cytoskeleton, and it moves things around. It kind of works like a ratchet to grab on and move things around. We have intermediate filaments. These are support for the nuclear envelope to maintain its shape. We have cell-to-cell junctions where our cells in multicellular organisms like us are held together, and we use these intermediate filaments to hold us together. And we have microtubules that are used for movement, and they're really, really important when we get to mitosis, to cell division. They form the spindle that helps to divide first the chromosomes, and then the duplicated chromosomes are divided between 2 daughter cells, and then all the organelles are divided up and the plasma membrane pinched in to get 2 daughter cells. So that mitotic spindle It's formed of microtubules, and the cytoskeleton moves everything around in that process. So that's what our microtubules look like. Our centrioles, we talked about these. These are those two barrel-shaped, perpendicular barrel-shaped structures. They're made of 27 microtubules arranged into 9 overlapping triplets. And got that pair of centrioles in animal cells. Those are called centrosomes. Plants do not have those. So here are my two barrel structures. And this is where I'm going to create and organize and run things as far as moving stuff around my cell goes. And then the cilia and the flagella I may have on some, but not all eukaryotic cells. Cilia are tiny hair-like projections, like little oars that are found around the outside surface of some cells, and they beat in unison to move the cell. And the flagella is a whip-like structure that spins to propel cells. So here's what again, that series of tubes around central tubes. We see that kind of similar structure here. And those are my eukaryotes. So hopefully, I think you all did really well on the lab for that. Just a little bit of confusion. Straighten out prokaryotes, eukaryotes. I hope this helped with that. Easy to confuse those first three letters, protists or protozoans versus prokaryotes. Remember, prokaryote before the seed. Protist is one of the kingdoms of eukaryotes. They're single-celled, simple, so they're the earliest ones. Protozoans, zoa, when you see that, zoology, study of animals. So protozoan, this is an early animal. So these are our simple eukaryotes, different from prokaryotes. Those are easy to confuse, so really stop and look at the words carefully. All right, that is it for Chapter 4. Thank you all.